மேம் அந்த ரெக்கார்டிங் வந்து ஒன்றும் சரியா ஓகே
ஸ்டார்ட் ஆயிடுச்சு சார். சார் நீங்கள் ஸ்க்ரீன் ஷேர் பண்ணிக்கோங்க சார். சார் நீங்கள் ஸ்டார்ட் பண்ணுங்கள் சார்.
Ma'am, can you see my screen?
Sir, the screen தெரியல சார். Screen is not visible, sir. Now இப்போ தெரியுது சார்.
Okay, ma'am. Good morning, everyone. We had a morning session about the drug target and analyzing of different drugs. Ma'am, can you see my slide? Is that clear?
Yes, sir. Yes, sir.
Good morning, students. I'm Dr. M.R.M. Sivasankaran, Assistant Professor in the Department of Biotechnology at Dwaraka Goverdhan College, Chennai. My topic is to discuss about the drug target identification and protein structure retrieval and domain analysis. So here we are going to see about the drug target and protein retrieval. Basically, it's about how we are going to dock the two different compounds like molecules, protein as well as the ligand. So the first thing is like we need to select for the protein which are like as a target to identify the diseases. If any disease or cancer cells will be having a signaling pathway which is involving a lot of
Participants, kindly switch off your mic.
Thank you, ma'am. So there will be a signaling pathway involving different types of proteins, which might cause in the different diseases and even in cancers. So it's like basically a target is like to find out the proteins which are causing the particular diseases can be identified with the databases, and with the help of database we can retrieve the protein and we can dock with the ligand of the molecule which is going to act as a drug. So optimizing that one, we need a complete database to retrieve the structure of a protein, which involves all the three-dimensional structure of a proteins to give a visible structure, virtual structure to target the disease-based proteins. So for that, we have our cells integrate various signals through a network of biomolecules that crosstalk to synergistically regulate the replication, transcription, translation, and other metabolic activities of a cell. So these are the proteins involved in replication, transcription, translation, and metabolic activities can be used as a target drugs, target proteins to inhibit the diseases. These network regulates the signal perception processing that drives the biological functions. The biological complexity cannot be fully captured by a single omic discipline because we need the integration of different types of bioinformatics tools as well as different disciplines like mathematics and other three-dimensional structure of a proteins is very much required for the 3D simulation as well as for the docking. So the lot of statistical data as well as the databases is very much important for the analyzing of the particular protein which are involved in disease. For example, if we have any diseases, that disease causing viruses or bacteria will be involved in the proteins which are involving in the diseases. So we can target the particular proteins which are causative agent for the particular diseases can be identified and target for the drug drug docking. So basically, the study of an organism in health perturbation and exposure to environmental disease is studied under the biological system. The bottle-up molecular approaches, genes, mRNA, protein, metabolite, etc., have laid the foundation of current biological knowledge covering the horizon from virus, bacteria, fungi, plants, and animals. So it's not only it's end up with only the diseases of bacteria alone. It can be we can take any n number of databases from the proteins which are involving in the even in fungi, plants, and bacteria and animals as well. These techniques provide a rather myopic view of biology at the molecular level. To understand how the interconnected molecules and components are formed and rewired in diseases, our exposure to environmental stimuli is the holy grail of the modern biology. Actually, we need to get the information, total information about the diseases which are in, and a lot of proteins involved in it, and we can target a particular proteins and we can inhibit the particular proteins resulting in the regulation of the pathways. So yeah, I'm just going to the next screen. Any doubts you can ask me in the between and we can line up and we can ask questions in even each in a slide. If you have any doubts, you can go ahead with it. So here we are going to talk about the drug discovery. Am I audible to anyone?
Yes, sir.
Okay. Yeah, we are going to see about what is drug discovery. Here we can see about a patient is having a particular diseases. The symptoms might be same, but the proteins or the pathways which involved in the diseases is might be different. So there should be a precision diagnosis for particular diseases. Nowadays we are going in for the general medications as we use it for normal drugs for day-to-day life, but in future it might be changed for the precision diagnosis based drugs. So for that, each and every person will be having the same symptoms, but the causative for the particular disease might be differ. And nowadays the disease caused by the bacteria is having different strains which are not inhibited by means of a present drugs which are available easily available in the available markets. So for in the future, we need to go in for the specific drug for the specific diseases which are like unique for each and every patient. So for that, we need to get an information about the pathways for the proteins, protein pathways or the signaling pathways which are involving with a lot of proteins in it, causing the diseases, or it might be a mutation causing the disease also. So in that case, we need to go in search of the genomics, proteomics, and other omics databases to find out whether we can find the particular proteins of the structure of a protein is available in the databases. For that, we need to go for the high throughput screening. The screening up to 10,000 compounds can be targeted for the against can be targeted against the target protein. For example, there will be a different databases that involving a lot of compounds which are easily available in the at present market. These each compound which not only inhibiting the particular proteins of a particular disease at present, but it can be applied to different proteins and find out whether it can be having a diversified inhibition factors for the particular drug. The virtual screening is very much important to find out whether the drug can be going to bind with the particular protein, and we can find its inhibition factor and its forces to find its attachment to its particular protein in the binding site. So there should be a combination of chemistry. That nothing but we need to find the drugs which are different structures. It comes under the chemistry and molecular modeling. The graphics and models helps to increase the activity. Later it can be done with the in vitro as well as in silico ADME models. For example, these drugs before going into the approval, we need to go for the preclinical test using the rats or mice or animals. right? So we can, instead of going in for the trial and error process of different drugs in the animals, we can prefer or screen the drugs which are more reliable for the drug targeting. For example, which can be inhibit the particular protein in this cancer pathway or the disease pathway, and the particular drugs, if it's going to more hits, can be used as a for the preclinical testing. So it will save the time as well as save the economically. It's more important for the drug industries to concentrate on the more specific drugs for the specific diseases rather than going in for the various applications of the drugs in the for part one diseases. So, how we target the drugs? So, actually, the drugs produce the therapeutic effects by producing the biochemical, physical changes in the target tissues of the organisms which invade the host. So, as I said, explained earlier, it's like the drugs which are going to change the particular effect on the particular protein which causes the disease in the host. In the starting, it can be in the human, it can be in animals, whatever it is, but it's going to be organisms. For example, if we are having a bacteria which causing a diseases, which cannot be inhibited by particular drugs which are easily available right now, like different types of drugs, like erythromycin and everything, because the strains has been developed. We need to go in search of the particular proteins of the particular strains which are causing the diseases, and we can inhibit the particular protein and we can cure the disease perfectly. So for that, the drug target is very much important, and the physical and chemical properties of drugs action on the drug targets, namely receptors, enzymes, carrier molecules, ion channels, etc. So, in the humans, there's a lot of disease caused due to this type of receptors. So, basically, targeting that particular receptors, the protein receptors, we can cure the diseases. So, the selection of target proteins, it's like basically 48% is like GPCR. It's a G protein coupled receptors. It's like 45% it can the drugs are targeting on this particular receptor proteins, and on the other 28% is on from the enzymes which are involved in the pathways, the hormones, and other factors like 11% it can be used as a drug target. And the ion channels, about ion channel proteins which are involved in ion channels, the gate pathways, it is about 5%, and the nuclear receptors is about 2%, DNA 2%, and unknown is like 7%. So the proteins are like basically we concentrate is like GPCR enzymes and hormone factors which are more convenient for the drug targeting to cure the disease in the human life. So the example of various receptors, the ligand-gated ion channels, example nicotinic ion channel, G protein coupled receptors, example adrenoreceptors, and enzyme linked receptors, example insulin receptors, and nuclear receptors, example glucocorticoid receptors. So these are different types of receptors which are helpful in targeting the particular diseases to inhibit the diseases. These type of proteins are used for the as a drug target. So what is ligand-gated ion channels like? Due to mutation, it causes like the sodium, potassium, calcium, and chloride channels, and acetylcholine-gated channels may lead to such physiological disorder as hyper and hypokalemic periodic paralysis. So these type of diseases are caused by this type of ligand-gated ion channels. So when we target the protein in this particular ion channels, we can cure the diseases by basically targeting the drugs on this particular proteins in this ion channels. So it's like the symptoms like Brugada symptoms, malignant hyperthermia, and myasthenia are the different types of symptoms which can be cured. Disease can be cured by using targeting this protein in the ligand ion channel proteins. So the next is nothing but G protein coupled receptors are the largest and most diverse group of membrane receptors in eukaryotes. Basically. These are acquired mutation genes encoding GPCR can give rise to a disease in humans. The anti-physiotic agents like clozapine and alozapine block the specific GPCR that normally bind dopamine or serotonin. So what happens like by blocking the receptor, these receptors disrupt the neutral pathways that give rise to symptoms of this type of diseases, schizophrenia. So these are different types of diseases can be cured by the targeting this G-coupled protein receptors. So next is comes about the enzyme linked receptors. Any doubts about the receptors, the previous slides? Okay, I'll go ahead with it. And the enzyme linked receptors, am I audible?
Yes, sir.
Yes,
Next comes the nuclear receptors. The nuclear receptors have the ability to directly bind to DNA and regulate the expression of adjacent genes. Hence, these receptors are classified as transcription factors. So, these glucocorticoid receptors are also known as NR3C1 receptors, which are expressed in almost every cell in the body and regulate the gene controlling and development, metabolism, and immune response. So, targeting these type of receptors also can be used for the development, like curing the development, metabolism disease, and immune response diseases also. So the other protein targets like specific membrane ion pumps, specific enzymes, structural proteins, cytosolic proteins, and other human cancer pathway interactive protein networks. So these are different types of protein targets which can be used for the some curing some diseases to inhibit the particular protein or to change the particular protein structures. We can cure the diseases. So here I mentioned about the HCPIN network. It's nothing but the human cancer pathway interactive network proteins. These proteins are involved in a lot of proteins are involved in the cancer pathway. When we target for curing the disease, if you're curing the cancer, we can target these type of proteins so that the pathways can be altered and the cancer disease can be rectified later on without using the drugs, without using the other type of normal drugs which are available right now. So the binding site is nothing but the region of a macromolecule such as protein that binds to another molecule with a specificity. So here in this structure, we can see the diagram. We can see the ligand is binding with the particular protein in the particular site. This site is a binding site, which it's like hinge or a site which is available, which can be used for the binding of the ligand to the particular protein. So this is the property where gives you the more interaction between the protein and the ligand will gives you the more enhanced result for the inhibition of the particular proteins. So another molecule specificity, the active site in the region of an enzyme where substrate molecules bind and undergo a chemical reaction. The active site consists of amino acid residues that form a temporary bonds with the substrate binding site and residues that catalyze the reaction of the substrate. So here this is the major part. We are just going to retrieve the protein from the database and the ligand from the other databases, and we are getting the three-dimensional structure of the particular protein and the ligand, which we can dock and see whether this particular having a compatibility or it's having an inhibition factor or not. So with this help, we'll get the results of reads intimating the drug is having an activity against the particular protein or not. We can identify with the help of the docking methodology. So here the drug discovery design is nothing leads into three different steps. It's one is nothing but identification, optimization, and development. So we need to find the lead is nothing but the drug which is going to attach with the particular protein site. Optimization is nothing but a quantitative structure activity relationship with the assays. It might be in vivo or in vitro. The development is pharmacokinetic studies of toxicology testing, formulation development, clinical trials, and regulatory approval. So these comes under the drug discovery. So each and every drug, when it comes to market, it will be causing all these steps, and the drugs can be used for particular diseases should be gone for the preclinical trials before going into the FDA approval. So all these things we need to channelize. We need to reduce the amount of spent by the pharmaceutical companies on the drug discovery. So basically, the docking is a one methodology which gives you the screening process and reduce the lot of implementation of drugs. More amount of drugs on a particular animals can be reduced. The characterization of drug target. The proteins are fundamental units of all living cells. It performs a large variety of biological tasks. The structure of proteins are much concerned, which determines its function. As I mentioned earlier, so the structure of proteins are much concerned, which determines its function. So we need to take the concentration of the structural database of the particular protein. So the three-dimensional structure of a protein determines its functionality as well as its the regions where the ligand goes and binds to it. The drug targets likes to be enzymes, particularly oxidoreductases, transferases, or lyases, and not ligases. They are involved in the binding, signaling, and communication. This can be summarized into key properties are desirable in the human drug targets. It should be what are the properties we need to find in the proteins are the high hydrophobicity, high length, motif present, no pest motif. Pest is nothing but the different types of amino acids like proline, glutamic acid, serine, and threonine. So these are motifs which should be present in the particular proteins as to choose as a drug target. and a signal protein motif also should be present. More than 2 N-glycosylated amino acids should be there. Not more than 1 O-glycosylated serine should be there, and should be a membrane location should be specific. So these are the factors which goes for the which we need to identify for the drug target of a particular protein. So here we need, we just came to know about different database in the previous slide. So database is nothing but where we get the three-dimensional structure of a particular protein or a protein of interest. So we need to go in search of that. We use the website, we need database called UniProt, the Universal Protein Knowledge Database, which involves PII, Protein Information Resource, as recently joined with the EBI, European Bioinformatics Institute, and SIB, Swiss Institute of Bioinformatics, to establish the UniProt. UniProt is a database from where we can type in the name of the protein, or if you have a unique ID of a protein, or if you want to find out the particular enzymes or proteins which are available in the particular organisms which cause the disease, we can go and type in the UniProt and find out retrieve the data from it. So here the UniProt, it comes from the it's a combination of different types of databases: as SwissProt, TRMBL, PIR, reference sequences, gene bank, EMBL, PDB, patent data, and other data. So here the combination of different database gives you the UniProt databases, which involves different types of data for the particular protein. If you want to search in a particular protein, gives the data of the total inbuilt data which has been provided already there. It gives you a detailed description of the proteins, what type of alpha helix are there, how many alpha helix, how many beta helix. So whether it's the protein is having different structures which can be utilized for the drug target. So every information will be provided. So the only thing is we need to target the protein which a protein of interest. Select the protein of interest, and based on the research after the outcome of the research, what we are going to what type of disease we are going to rectify or we need to solve. Here are the different types of peptide search also. Peptide is nothing but it's it can be a monopeptide, a dipeptide, tripeptide, or tetrapeptides. Different types of peptides are there, which can also be used as drugs and can be rectified from this particular databases. There is starting with the protein information resources, from where we can get the three-dimensional structure of the peptide, or it can be a peptide, a singular linear chain of a peptide can also be isolated. So here the different sites, it gives the information about the proteins which can be rectified from the particular resources. So here we get information about the output of the particular inquiry over here, the protein name, and we're getting the different days. So these type of database search and retrieval of the proteins and everything will be in detail, will be explained in your practical session. I'm just giving you about the introduction about what are the different types of databases available and what's the unique properties of the particular database. The UniProt protein record will be involving a lot of information. It's about the unique ID, or it's having its homology of a particular protein can also be found from this database. Homology is nothing but the same protein structure, or it's having different functions, or different functionality will be having the same, different structures will be having the same function, as well as different structures causing the different functions also can be different types of proteins can all be isolated in this type of databases. So here, now we come into a topic also how we are going to get a database, how we are going to search the database for the protein retrieval. So basically for that, we need to identify the biological origin of disease. What is the disease, and a potential target for involvement in the first step is the discovery of the medicine. As I explained earlier, discovery of the medicine is basically depends on the uniqueness of the protein which are causing the diseases. It can be either the protein can be either from the disease causing organism, or it's proteins which are involved in the cancer pathway, or protein involved in the receptors, what I said in the previously about the protein receptors constituting disease constituting due to the mutations. So here the objective of the particular protein structure retrieval domain analysis is nothing but to study about the protein and its structure, to introduce the PDB and its importance, to learn how to retrieve the structural data of a protein using a PDB database, to describe the PDB file format. So here, a protein database contains the information about 3D structure of a protein. One. The next is PDB file format is a standard file format for protein structure files. It describes how molecules are held together in a 3D structure of a protein. The PDB files also contain information about data collected, molecule name, primary, secondary structure, ligand, atomic coordinates, crystallographic study factors, and NMR experimental data. Each entry in the PDB is provided with a unique identification number called PDB ID. It is a 4-letter identification number which consists of both alphanumeric characters. The structural information of a protein can be determined by the extracrystallography or a NMR. It's nothing but nuclear magnetic resources spectroscopy method, which gives you the structure as well as the patterns to the different types of patterns available in the proteins. The X-ray are X-ray diffracted by the electrons of complete size atom, resulting in a patterns obtained as small spots in X-ray films. These patterns are used to calculate the coordinates of atoms in the protein. So to identify the atoms in the proteins, it's XRD, and NMR gives you the structure of the molecules. Database derived from the PDB. The PDB will be given type of different types of database that has been retrieved, derived from the particular PDB. Is nothing but the scope is nothing a structural classification of proteins that groups different protein structures. HSSP is nothing but the homology derived secondary structure of a protein for 3D structure and a sequence of the protein. And a CAT is nothing but for protein structure classification according to the evolutionary factors. So from each and every database giving you the unique output of the particular proteins. So we can use the different databases for the different usages. Now comes the protein database. To retrieve the structural data of a protein using a protein database, open the browser, go to the PDB homepage. Here we can find in the PDB homepage, we can find the molecules of a protein molecule by typing in the name of a PDB ID in the box provided. It's over here. We can find a box here. So here we can find the PDB ID and we can go in search of it. For every structure in PDB, there is a unique ID. It will be 4-letter alphanumeric as I explained earlier. And it will be, for example, it's like 1HES indicates a protein named PI3 kinase. So each and every protein should not be like proteins will be having the same different IDs, same ID. We have a different ID for the uniqueness. Even a small changes, it can give a different IDs. Also, there is option search for the ligand, sequences, macromolecule, author, and all categories. The PDB format can be called as a standard format of a protein structure files. Any doubts up to now? Students? Okay, here we go for the search box in a protein database. When the protein name is provided text box, it results in displaying showing the data like molecule, name, PDB text, structural domains, ontology terms, and particular information of the molecule is obtained. So everything will be provided in this output. And next is like a search can be changed by the quick search IDs and keywords in this next column. And we can change the different search and find it out whether different proteins can be retrieved. The hatch search criteria option will add an extra box to search the query type as shown. So we can add, for example, if we want a protein with this particular type of bond or different types of liquid structures and everything, we can go in search of this extra search query. So next is like example retrieve sequence data. If the kinase is showing that, I'm showing just giving you the example like how you type it. So here we have EGFR kinase domain which is going in search for the particular thing, and this is the output we get. This output is like we can get a literatures and a journals, a PubMed ID, primary citation of related structures, and other abstract of research publication. Everything will be in detail will be produced as an output. So here macromolecules, and macromolecules is a type of output which we get from the particular database for the protein sequence ID we have inserted. So in the macromolecule, it describes about the protein classification, molecule total, structure, weight, chains, length, organisms. In this case, it is a transferase inhibitor, which has been used for the inquiry. And small molecules, nothing but the ligand, which can be retrieved from the databases, which gives you the 2D diagram interactions. The ligand should be having a less molecular weight. So based on the criteria, the ligands are selected for the targeted proteins. Gives you the idea about the structure of a ligand. So here the examples of ligands nothing but folate, transferrin, galactosamine. So the target for the folate is folate receptor, transferrin receptors is transferrin for the transferrin receptors, and galactosamine is receptors on the hepatocytes. So these are like a tumor target ligands. So these are basically goes and target on the proteins solved in tumor pathways. So experimental data and validation is nothing but gives you the overall information about the like X-ray diffraction, NMR, etc. It is used to obtain the structural data of particular protein. The structure is validated using a different methods like Ramachandran plot, R value, and is shown under the structure validation column. So here we can get the information about the Ramachandran plot of the particular protein we are going in search of it. Entry result is history, like previous history, whether it has been given a search has been done or not on the particular protein, or it's a new type of protein. Everything comes under the entry history. This is how the three-dimensional structure of a protein will be for the particular enquiry. Is like a EGFR kinase protein, which has been utilized for the view of three-dimensional. So clicking the option like 3D option, we can get the protein structure of the particular enquiry. So here we get the three-dimensional structure of a protein, which we can rotate and find out its properties by finding its structures for the binding factors where the particular ligand goes and binds to it or not. So the FASTA format is like gives you the information about the total amino acid sequence for the particular protein. The FASTA sequence can complete represent the protein sequence. The FASTA format is the simplest sequence format, which starts with the open bracket. It's like a greater than sign symbol, followed by the unique ID. Other comments are computationally represented as protein sequences. So this is the FASTA format, which gives you the information about the amino acid sequences present in the particular enquiry protein. So here are the different types of output we get from the protein search. It's like header, title, command, source, keyword, EXP data. Everything is involving different types of information for the particular protein we have searched for. For example, header contains the file name, date of submission, molecular PBID, and everything. Title records like what type of a PDB entry has been given for the title of, for example, ID for the particular protein. The compound record includes the protein name, and the source record contains the name of organisms which a particular protein is obtained. The keyword records contains the functional classification, metabolic role, biological chemical activity, and structural classification of the particular protein. And EXP data record contains the X-ray diffraction, electron crystallography data records of the particular protein we are searching for. So other things, the same thing. The author gives you the information about the author, the name of the contributors of authors which are involved for the particular database, and other records are like in general. So it's a huge type of output is given for in detail, like in a master record, it's contained about the HET records, the number of helicals records, sheets records. So we can get a full information of the proteins which we are going in search for it. So we don't need to find out like the data is missing or not. It's everything has been updated eventually, and it's given the present scenario of a particular protein is also updated, which gives the information totally for the ligand binding site and used for the drug targeting especially. So here we are going next tomorrow we are going to talk about the docking. So the previously we need to know about what is target. The protein is a target, and the ligand is a drug. We are going to goes and binds to the particular protein, whether going to inhibit the particular protein or not. The selection of the target depends on your research, whether it can be a drug protein from the disease-causing organisms, or it can be a protein from the human cells which are causing diseases. Anything. The ligand is a drug which we are going to go and binds to the particular protein. So the selection of the ligand should be is very much important for the particular proteins. Thank you very much. If any doubts or something, you can ask now. Okay, ma'am. I think so. There's no questions. I think.
Sir, chat box we have question.
Sorry, ma'am.
In chat box we have the question, sir.
Yes, yes, ma'am. Okay. So here, I just got a question from the student, like where can I get the monoclonal antibodies and everything. The proteins, we can use it like a UniProt and other things. We can get the information also about the monoclonal antibodies updated eventually. You can get it from the UniProt. You can get from the other databases which are available in the same PDB databases. Specific for particular monoclonal antibodies also, it's also available in the PDB databases. Any more questions? Actually, the first question is like how to upload a new sequence in the kind of database. So here it's like if it's the database will be giving the information of the sequences is already present or updated. So if you are giving a new sequences, then you can upload it with the help of the database. You can publish the new sequences which has you have been isolated if it's going to be new sequences, it can be published by the particular database. They are giving the information about that one online itself. How to choose the effective ligand? Basically there's a lead optimization. It should be a low molecular weight, as well as it should not cause any side effects. It should have a good binding properties with the proteins we are targeting for, and it should not. It's like the less molecular weight is the first criteria we need to check for the effective ligand. Any more doubts? Thank you. Yes, the binding site selection and everything will be done in the molecular docking session. Since I just given about introduction about the selection of a protein, you might have a confusion like whether it would be a huge step for you to go ahead with this session right now. So in the docking session, it will be clearly explained about how to select the target proteins and how to find the site of the particular proteins, and which site we can dock the ligands, and everything will be discussed in the docking session, molecular docking session. Thank you. And one more question is like how to identify the target protein. Yeah, target protein is based on your research. It's going to be like a cancer-based studies. Like you need to go in for the cancer-causing pathways, signaling pathways, where the proteins are interlinked with each other, causing the cancer. For example, apoptosis is not occurring, then it's due to the presence of the or absence of the particular protein, or inactive protein might be there. So by inhibiting the particular protein which is causing the cancer, also we can cure the diseases. So it's a one type of research where we can find that protein target. Another type of research is like if a disease caused by particular organisms which is not inhibited by the drugs which are present, we can go in for search of the proteins, three-dimensional structure for proteins, and we can try with a different target, different ligands, and we can find which ligand is having a more effective against the disease-causing protein, and we can use a molecular docking methodology for that, and we can find out whether it's having inhibition factor or not. And then that way we can target the proteins. Yes, you don't need a software for the selection of a protein. As we retrieve the data from the protein databases, the same way we can retrieve the three-dimensional structure, I mean the 2D structure of the ligand, which is available in the database, the ligand database itself, which will be discussed in the molecular docking. What type of database for the ligand selection as well as the protein selection will be in detail, will be explained in your docking session. Especially if there's no software for selection of that, it's a database. We need to select the structure, and that structure can be uploaded in the tools what we're using for the docking, both the protein and the ligand. I'm sure that is there any more questions you can ask now? I just answered most of the questions. If not, you can ask the questions in the chat box right now. All right, hoping that there's no more questions, and even if there's a question session, we can ask in the practical session, or the tomorrow we'll be having a molecular document introduction class as well. It will be explained in detail about the protein structure identification as well as the ligand selection in that. Thank you. Thank you, ma'am.
Thank you, sir.
Thank you for your enlightening session, sir. Participants will have a lunch break and join the next session by 12:45 p.m. Thank you.